Let's introduce briefly the different participants. So we start with Dr. Dashdorj. She is a co-founder of and head of the Onom Foundation in Mongolia that has played a very important role in shaping the public policies on hepatitis C, hepatitis B, and obviously hepatitis delta in Mongolia. And it is important to underline that it was really a big achievement to transform activity into policies, and this is very, very important. Pietro Lampertico, I don't know whether he really needs introduction, is the professor of gastroenterology in Milan, and he leads the department of the Center of Liver Disease and Pathophysiology and Transplantation, University of Milan. He's been a leader in hepatitis B and hepatitis delta lately, clinical research, and I think I have not to say anything more. Professor Njouom is the director of the International Network and the head of the virology department in the Institut Pasteur in Cameroon, and he formed in France. He has a PhD in addition to the medical degree, and he has been part of the development of WHO guidelines for viral hepatitis as well. And finally, Fabien Zoulim, that is like me in Lyon, is a professor of gastroenterology in Lyon, is the director of a research unit of viral hepatitis at the INSERM here in Lyon. He founded the Lyon Hepatology Institute, and I think I have not to underline more his contribution to hepatitis B and hepatitis delta research. Having said this, I would start asking the four panelists to eventually talk one after the other and point what was for them relevant to discuss in the three presentations we have, while Maura can select the first one or two questions from the chat and the Q&A section. There are several, and then eventually I will ask myself some questions. But I think that the most important point is the panelists trying to challenge our speakers. So let's start with Dr. Dashdorj from let's say the Mongolian perspective, or the Asian perspective, or the Delta perspective in a highly high prevalence country.
Thank you for the nice introduction, and thank you for all the nice presentations. This was really wonderful, especially having the WHO. That makes it really nice. So as you all know, that Mongolia has now I think a lot of papers actually came out really showing that Mongolia has the highest burden in the world. So we are really having issues from the testing to the treatment, and treatment is being one of the really urgent problem. And I also see on the questions from Kyrgyzstan asking very similar questions. We have at least a drug two drugs that does something, especially the bulevirtide obviously makes great change, that it's especially not having a lot of side effects. Despite its being a long-term treatment, this really makes a huge difference. And we would like to have the drug on the ground, but we are really struggling to get it. So just to put it in perspective, so the Heiner did really nice presentation, and then one of the thing really given me like quite difference was that Heiner was saying on the presentation, on the slide, that with the 176 patients, huge data set. But I can tell that in my clinic, it's a small clinic within Mongolia, we have 4,300 patients who have HBV RNA positive. They need treatment urgently, but we do not have treatment. And now we have some drugs on the ground, but that the pricing is that with Hepatitis now, official pricing is $2,500 per month, whereas the income of Mongolian people is around $300 per month. So for 1 month treatment, you need to earn almost 10 months salary. So that's just does not work. And there are some negotiations going on. But what we hear is that with the negotiations, it's only coming down possibly to $1,000, which is again 3 months salary. It's not affordable for most of our patients. So I wanted to ask Nicholas, is there any plan for WHO to kind of facilitate and to have some sort of a framework to make these drugs available for these patients, especially because we do not have any other choices? The country like Mongolia, for example, government put some money aside. Now we are purchasing for the first time 250 patients peg interferon. But that's for the first time. So it's still some positive movement, but it's not enough. So that's what I wanted to ask. The second question for the Nicholas's presentation was that also Elizabeth's presentation, there are a lot of this new testing diagnostics are being developed, but if you go through all of the pre-qualification procedures at the WHO level, it's very, very slow and very bureaucratic process. Do you have any plans for making it, especially for hepatitis delta, do you have any plans for making it a bit more expedited, like orphan drug, similar type of orphan testing, approval process, etc., something similar that utilized in the SARS-CoV-2 epidemic? So these are the two questions that I feel that probably needs to be asked. Thank you.
And just before Nicholas answers, there are also some questions in the chat that went into the same direction. What can be the strategy to allow access to these drugs that seem to interferon is a little bit a different problem, but for bulevirtide we really saw the implications of the role in treatment. But what are the strategies? What can be done? Are there discussions at which level? And eventually also John, if he has elements to add, perhaps would be nice to hear from you.
So I think it's for me to start. Is it right, Massimo?
Yeah, if you can.
I unfortunately think there's not a very easy answer to that. I think I would just remind everyone in terms of, WHO cannot be engaged in direct negotiations with manufacturers of it being diagnostics or drug companies. I think what we generally contribute to, let's say, the space is that we have recommendations and that we can be part of partnership efforts to bring pricing down or highlight efficacy and awareness around certain issues. So I think the first thing here that we all have to do, that doesn't respond to that question, but is to continue to increase the advocacy and awareness about the importance of hepatitis delta. All our presentations pointed to that. Now, in terms of drug prices, it is really mostly to the countries and the negotiations with the pharma companies. Here for bulevirtide, we're talking about one specific pharma company to negotiate those prices. And I think they should depend on the income level of a country, right? And the margins and for the company and the prices for the country should potentially vary in low and middle income countries. Now, if we're thinking back to hepatitis C, for example, where we started with incredibly high treatment prices, you know, I think as well, one thing that really contributed, that wasn't the only effect, but was to put pressure on the companies who were producing those drugs on highlighting those from civil society, patient groups, countries, highlighting the importance of making this more widely available and not only in high-income countries. There is now, to finish, and then good to hear from John, there is actually a mechanism in Geneva based that is not linked to WHO, which is the medicines patent pool. And the medicines patent pool can coordinate with pharma companies around voluntary licensing. So that would be a mechanism, you know, where a company grants for a specific drug a licensing mechanism for generic manufacturers, for example, to produce, if the drug is still under patent protection, to produce that drug for access prices. And everybody possibly knows that this exists for hepatitis C, for example, and several combinations, and covers more than 100 countries for sofosbuvir, daclatosvir, for example. So that would be an existing mechanisms, but I don't know that bulevirtide, we are there already, you know, in terms of what we know about this drug, how much it costs, the engagement of the manufacturers to make this mechanism available. But we should have this discussion with MPP.
John, do you have any news about because now the mechanism?
I don't have any news.
The important point is something going on.
I just want to champion, you know, many of Nicholas's comments. I mean, I think we're, you know, the evidence is growing in strength to have a compelling case for why this drug needs to be available in countries such as Mongolia, and then we can begin to, you know, get MPP involved and speak directly to the manufacturers. You know, Gilead has an excellent record of making their medicines available via generic manufacturers. So I think it's a great opportunity to develop a public-private partnership, and the coalition can definitely, you know, have a role in that. So I, you know, look forward to seeing how we can solve this problem over the coming, you know, several years. It's going to take a little time, but I think the evidence is really strengthening the case for why there needs to be more affordable options for high-burden countries such as Mongolia. I would like to think, Nara, that you know, the move of the government to support some hepatitis delta testing and treatment is a positive step forward. Just they're beginning to hear the message that delta needs treatment. So I think I see that as a positive step. Do you agree?
Absolutely. This is a positive step to the right direction, but it needs one of the really big problems with the hepatitis delta patients is that they are really so quickly progressing, and they are the people who are actually very young people. So I think we need something not in years' time, hopefully within a year or two.
Okay,
so
I hear you.
Are really needing it very urgently.
No, thank you. Let's move on to the other comments. I don't want to have
Before going to the some more medical or clinical questions, let's hear something from the perspective of Africa. What is moving around there? Whether there are specific questions from Richard.
So thank you, Massimo. Thanks also for the organizer for the invitations. And Amazonia seems as Africa, because most of what she said regarding Amazonia is the same thing in Africa. But particularly in Cameroon, where I'm working, because I'm working in the National Reference Laboratory, where we have to confirm most of the people who are suspected to be infected by hepatitis delta infections. All the analyses are centralized. So people that are infected are mostly located in the remote area. And if they want to know the status, they have to travel from the remote area to the central lab. And I think that most of the big challenges is the testing. Testing also is a very big challenge because I think that I have one question. So let's say Elisabetta, or it can be the same question for Dr. Nicholas, to see what is the challenge in developing rapid diagnostic testing for HGV. We saw that for HIV, for example, after 10 years of after discovery, we have many RDT testing, but now for HGV, 50 years after discovery, there are no commercialized as HGV. And the big challenges now for us is to decentralize the diagnostic so that because infected people are found in the remote area, and which is very nice for us to, let's say, decentralize diagnostics so that the testing could be done locally and we could have the results at the center, the result at the central level very quickly and see how they can take care of them. One of the second thing that I can think about is the data, the epidemiological data. I think that most of the epidemiology in Africa are underestimated because the diagnosis of HGV is not done routinely. And most of the data that we have is just some sero survey that we did, for example, during, let's say, demographic health survey. The one that I published 2 years ago is the data that is used, but we are in an endemic area with people that we have more than 10% prevalence of hepatitis B, and about 10% of HBS positive patients are also HGV positive, and we have let's say a disparity in the prevalence regarding the geographic area where the people live in. I found more than 50% people HBS positive, whereas Delta positive, those are living in the forest area in Africa. So in summary, we need testing to, let's say, to give good data, and the data is very important if you want to think about eliminations, and also it's good to develop simplified and rapid diagnosis that can be used for decentralization. That's what I can say. Thank you.
So I would ask Elisabetta to provide a very, very short answer if she has a contribution there, because we want to hear from Pietro and Fabien and Einer, something specific that I will point out in a moment.
So thank you for the question. I think that the key point that prevented up until now the development of technologies, assays, was the lack of a definitive approved therapy. So with the field of drugs moving, I also think that the field of assay and diagnostics is still moving. So many companies are involved in developing new assays, and this will also in turn result into pushing development all of testing and also user-friendly tests. So this is my short comment.
So there is a long way to go. I think if the others want to do some fast comment on that. But the question for Pietro and Fabien and Einer eventually, and after they can do the comments as well, is: do you think that there is hope to have bulevirtide as a generic? Because there are two situations for me. One is the chemistry, whether it will be easy to go for that, first point. Second point is for hepatitis C, everything was easier because it was a finite therapy that was having 90% or more success. So let's have the trio of the clinicians and virologist clinicians there. Just give us something to think about on how can be the development of more access to the existing treatments. After we have the new things we heard from Einer, we can discuss hours about the side antibodies and the rep, but we will the NAPS, we will not do this today, probably. We have no time. So perhaps Pietro want to start, or Fabien? Okay. Fabien start. And then we will go to the...
Okay, so I'd like to thanks for the invitation and congratulations for the speakers. So my short answer is no. I don't think that the drug will be easily available as a generic. To my understanding, is a kind of complicated chemistry. So no, the answer is, my short answer is no. But I have one question for Heiner, which is related, of course, to clinical issues and stopping rules for bulevirtide monotherapy. Any suggestion?
That means how long we have to treat, in a way. That is linked also to the development and
Exactly,
of the market.
exactly.
I don't know whether we should now spend 3 hours to discuss the stopping issue, or whether we first should
You have 90 seconds.
No, but maybe let's first finalize the other. Pietro was very strong in saying that there will be no generic, and you asked the question, Massimo, on the chemistry, etc. Actually, in the end, it's easy. It's a peptide. It's a peptide with a little, let's say, modification, the mustulization. So it's no miracle to generate this peptide. So I was, even before bulevirtide was approved in 2017, 18, when I talked to patients, at that time I said, if I would be a patient myself, at that time I said, I would just ask the company in wherever to synthesize me the peptide. I would mix it myself and would inject it myself at that time. It would be life-threatening, because theoretically, it's not that complicated let's say, produce this peptide, but obviously there are patent issues, developing issues, administration, formulation, and you all may know that we don't have the approval by the FDA yet, not because of concerns regarding efficacy, but regarding all this production changes and quality, etc. So I don't know details, but it's not that the FDA was not convinced by data. It's all these QM issues. So I cannot comment on this. I have no clue whether there's a company starting things in parallel, but theoretically, in my understanding, it's not the issue, right? So
let's just make a comment, or let's hear from on the same issue from Fabien, if he knows something more or can add something different on the chemistry, and then eventually we will make one comment on that, because I think that is relevant.
No, okay, so thank you, Massimo. No, I don't know more about the chemistry, but I think we need to first to figure out pragmatic issues for the already existing compound, and I'm not talking about the generic. So there are issues about storage, as Heiner was alluding to, issues with the reconstitution of the product every day, and the everyday injection. So there's a lot to be done if we want to improve what we already have, to have like a long-lasting drug, bulevirtide, with a pre-prepared syringe. So that would make already a huge change, and then we could think of generics and so on. So the other point to look into the future is that there might be some newer version with small molecules inhibiting viral entry that are in development. So it's a future. So this might be in the end the option to have just a pill, just a single pill, instead of a peptide, which has all the issue of storage, and stability and so on. So it's just a different perspective, but we know that there's a problem.
Yeah. Coming back to the chemistry, doing peptides is easy. The problem is to standardize. The modification is feasible. It's easier to modify one thing than build up a very complicated chemical structure for some of the compounds we see going around, like for an example, the NAPs. But on the other hand, what is very complicated is to standardize and to have a very, very well-done purification of the peptides. This is what the machine is easy. I mean, even a small startup can do it, but then if you have to produce a big quantity, would have been nice to have Stefan here to comment on that, Stefan Urban. But because they worked a lot, obviously, on that. But the point that I see in enlarging toward a real generic, I think, is that different is licensing. If Gilead wants to go a certain point into that, and this is what probably the coalition and all the advocacy group should do, is to enlarge the access via a wise policy of licensing versus direct access to the Gilead drug at special prices. But this will be a long road, and I think the clinician, we have a few minutes for the last round, perhaps the clinician can just comment on the last point. How long? When to stop? What is there that is coming out from the data, the newest data? We have all these real life cohorts and all that say we have X number of patients, we stop it after this time, this is what's happening. What do you think? Let's give to the audience a kind of perspective today, because probably in 3 months we have new data.
Pietro, you should start.
Actually, the Heiner, the question was for you, so you have to start.
No,
you can ask Fabien to answer, but would be a job.
No, I think the point is, quite frankly, so obviously I wouldn't stop a patient that still has 500 units of HDV RNA. So ideally, the patient should be not detectable for HDV RNA. The question is whether there are very low residual dips of HDV RNA detection indicate really replication needs to be answered. But the data that we have seen thus far, in principle, this is possible. But the very big buts. I think this is things for clinical trials at this stage. There will be post-treatment flares in individual patients. Fortunately, in the clinical trials, there was no major, let's say, liver failure associated with flares, but we haven't seen data on 500 patients stopping. And if in 500 patients stopping, there may be 5 patients who really develop a problem. And therefore, I would be, at this stage, very cautious to stop outside of clinical trials. I understand that in distinct settings, resource-limited areas, you may not have the option to continue forever. And therefore, we need this data really urgently. It's not something of science that we're playing around. We need this data for safety of patients if they cannot afford lifelong treatment.
Pietro or Fabien, do we have something in the initial response in terms of RNA that can guide us to say that the patients is more likely to stay on response? Let's say, how fast you get non-detectable RNA. Are there data for that to start to build up on this, or in your experience there is nothing from your cohorts, from your trial?
I can make a comment for bulevirtide monotherapy. I think Fabien has a very good data on the combination. So for monotherapy, I think that yes, the earlier you get PCR negativity, the more likely you will maintain PCR negativity. And then the more likely you will have a patient with like 2 years PCR negativity, and eventually he or she could be a candidate to stop in therapy. But unfortunately, the number of patients who achieve target not detected by week 12 or 24 on therapy is very, very limited. So if you want to reach 2 years PCR negativity, then probably you need to treat hundreds of patients for 4 years to get some with 2 or 3 years PCR negativity. That's monotherapy. Fabien, you have very good data on combination therapy.
So with the combination therapy, well, we have to, with the caveat that the number of patients is not huge. We have to be very careful. So we have data also suggesting that the earlier the RNA is undetectable, the better it is, and so that the undetectability of RNA should be as long as possible before stopping. The other point is that what was shown in the combo study is that if RNA is detected
below the level of quantification, then there's a risk of relapse. So again, so this come back to the point that was discussed. We don't have time today on the assays. The robustness of the assay will be extremely important if we want to have a response-guided treatment, and that's a must. We need to improve the assay and standardize the patient monitoring.
Maura, I give the word to you.
Yes, so at least the good news you can say from the study that we have all seen, it's somehow that if we don't see for certain time in the blood the virus, at least with boulevarditis, it seems that there is also no more there in the liver. The question is how long you have to be negative to be quite sure that you have good chance to be really negative or to control the disease. So it's a very difficult issue. Unfortunately, we didn't have really much time to discuss so many aspects. So we will need more, I think, of this webinar and opportunity to discuss, because it seems that we need to act together, I think, to increase a coordinated screening strategy. If we don't have a screening, we don't know what's the burden. If we don't know the burden, at the end, we will not know how it is important that we keep developing better drugs and how to apply and to implement, and with need to be approved also for boulevartide, not having FDA approved, still it's some problems, some issues, and I think we need better drug, especially for the endemic countries. So this is a discussion that we cannot end here or resolve, of course, here. But it's something that I think we are, all these coalitions and people and company needs to be aware of and keep working on that, because people really suffer of this disease. Even if, Einer, you mentioned, we have interferon, it's not always bad. It does something, but the hope, we still need interferon to be used in combination. This will be also a topic of a symposium that we will have in Chicago about trying to understand the still ongoing utility of interferon that also organized from ICE-HBV. So there is a lot to discuss about B and D and the good of having still interferon. So we need to work a lot.